Forget frequently asked questions, common sense, common knowledge, or Google. How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5% go above and beyond and become very good at what they do. But only 0.1% are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in fields such as sleep science, cancer, stem cells, ketogenic diets, and more. Get ready. Here come the geniuses. This is the Finding Genius Podcast with Richard Jacobs.
Hello, this is Richard Jacobs with the Future Tech and Finding Genius podcast series. I have Professor Aaron Kaplan. We're going to be talking about ecological processes in photosynthetic organisms. It's going to be pretty niche stuff. One thing we were discussing offline is desert biological soil crusts. So we'll get into that and one other issue. So Aaron, thank you for coming. How are you doing today?
Not too bad. Hope the same for you.
Yeah. Well, first, a few definitions. What is a biological soil crust? What's the job of it? And what does it look like? And what does it do?
Well, when we walk on the dunes in the desert, we do see something like a crust covering the dunes. The thickness of the crust depends on the activity of the organisms. Basically, it is formed by a filamentous cyanobacteria, also frequently named blue-green alga. They excrete polysaccharides that adhere to soil grains, so that you get a crust kind of. Now the crust is physically is very complex, though it may look very simple. It is complex with what I may refer to as tunnels of moving light in, breaking the light into the crust and changing the local environment. This is essentially one of the harshest habitats on Earth. The productivity there is quite high depending on the time. Now, the organisms undergo very frequent, daily, almost hydration-dehydration cycles. And it is the vegetative cells that undergo the hydration mostly by early morning dew and then desiccation. The soil temperature can easily reach 60 centigrade or so, and they really dry out almost completely. So we looked at the mechanisms that this organism acquired to essentially proliferate in this environment. And among the other organisms that we isolated is a small green alga. Now we often tell the students that organisms that acquire the ability to cope with stress, with a certain stress, they do pay a price. It is in the textbooks, for instance, that they grow slower and perform much slower than other similar organisms under optimal conditions. But the green alga that we isolate.
Oh, quick question. What deserts? Is this like the Negev desert or what?
Yes.
Okay.
This one was isolated right between the Israeli, close to the border between Israel and Egypt. There is a dune area there that we isolated the organism from. Now,
what kind of plants grow there that use these crusts? Like, so the crust is not coming from the plant itself, but it's coming from symbiotic organisms, or like who's making the crust? Where does it appear?
The crust is formed, as I mentioned, by the cyanobacteria, but as it develops and it gets thicker and thicker, more and more organisms are there. Plenty of different heterotrophic bacteria that make their living on the products excreted by the cyanobacteria. The cyanobacteria is the pioneer and the main primary producer. It is the guys that make the job. They produce the carbon skeletons. They produce the carbons used by other organisms. They fix nitrogen, and they form the proteins. So everybody else is making its living on this cyanobacteria. But what I'm
Look
adding
like?
to,
What does the crust look like versus? Is it all around the? So is a top layer of sand around a plant all composed of crust, or what does it
not
look
not
like?
not only around the plants, all over. There's a large variability in the structure of the crust and the large variability in the organisms inhabiting the crust, spatial and temporal. So it is found in all the deserts around the world. It covers quite a bit of space, and it is considered to be quite productive in some parts of the world. Yeah.
I
Now,
think that there's just bare desert, but the entire surface of the desert actually is composed of a biological crust.
it depends where the importance of the crust, socioecologically speaking, is that it raises the shearing energy required to move the sand, and consequently, it is the more developed it is the best way to combat so-called desertification, for instance, which is the movement of the sand in southern Sahara, for instance, and leading to large migration of people because of the destruction of the crust. This is a very well-known phenomena that when the crust is scrambled by the herds of the Bedouin or other people, you get far more sandstorms all over the world. In China, for instance, in Beijing, there are quite a number of days that you can hardly see somebody standing a few meters from you because of sandstorms. And the Chinese spend billions of dollars in attempts to stabilize the crust, and they did pretty well job by spreading the cyanobacteria all over the country in the desert there. It's a huge effort was done there.
These crusts initially form.
As I mentioned, it is formed by the adhesion of the sand particles to the polysaccharides excreted by the cyanobacteria. That's
Assume that there's nothing living except maybe a few stray animals in sand desert. It seems like there's just nothing there.
No, there's a lot there. There's quite a number of different bacteria and classes, and also quite a number of animals there. But what I'm adding to is something else, which is, I think, fascinating, because over there we isolated a green alga that it is the fastest growing photosynthetic organism ever reported. It is, if you grow some plants in your balcony and it gets some sun, it gets burned. This is a process so-called photo damage, photo inhibition, photo destruction, whatever. And if you read the literature, the people that studied it for the last 50 years or so would tell you that it is an inevitable process, destroying one of the photosynthetic reaction centers. Now, this chlorella is completely resistant to photo damage, and it took us quite a bit of, convincing our colleagues that this is in fact the case. It doesn't undergo photo damage, no photo inhibition, and we even took genes from this chlorella and placed it in a model plant, higher plant, making it far more resistant to photo damage. So here is an organism isolated from harsh environments that can do something for the benefit of mankind.
So when you say it's a very fast photosynthetic organism, how fast, and how do you determine speed? What's the metric?
You measure the growth rate, and you can measure either the number of the cells, the amount of protein. You can measure how much chlorophyll accumulates. Chlorophyll is the green color of plants that absorb the light energy. You can do quite a number of different things. You can measure the amount of DNA. Quite a number of parameters. Generation time of this organism is 2 hours or less. It is as fast as a budding yeast that people use for various purposes. If you compare it to other algae, for instance, a model organism often used, so-called Chlamydomonas, its generation time under the best conditions is between 6 to 8 hours. In algal growth facilities, you normally get one doubling a day, and this chlorella that I named Chlorella Wadi after Professor Yitzhak Oad, a dear friend of mine and one of the pioneers in photosynthesis research. That does 4 to 5 duplication a day. So in algal growth facilities, it is quite fascinating.
Some of the uses, you said you've merged it with a higher plant. Like how much more efficient was the plant, or how much faster did it grow?
Well, we didn't go so far to how faster it is, but it is far less sensitive to high light intensity, to high illumination. And that some may take over to crop plants, and in the future I expect it to be used there, and to reduce the sensitivity of crop plants to high illumination. You know, if you take a corn plant, then when the plant develops, the top layers, the top leaves, they experience a very high light intensity. Then they get shaded by the next generation of leaves produced. But originally speaking, they're exposed to very high illumination, and then they get into a shaded area covered by other leaves. So the plant must be quite flexible to change the photosynthetic machinery to adopt the changing light intensity, and there has been a lot of studies on these aspects.
Well, in growing conditions, I know they vary tremendously around the world, but how often is there a situation where the intensity of the sun is too much that it starts to destroy the canopy?
Oh, it's well, depends where you are, but in large parts of the globe, the light intensity is far higher than the plant can deal with, and there is a destruction of a so-called protein D1 at the center of photosynthetic reaction number 2, and this is number 2, though it is the first one that kicks the electrons. It is named 2. I often say that it is only to confuse the students, but destruction of this reaction center is a major cause of decline of the rate of photosynthesis and growth under high illumination. Yeah.
So have you gotten to the point where implementing this into a plant, does it have different growth characteristics? Does it just form, an initial set of canopy leaves and it doesn't form subsequent leaves below it? Or?
That wasn't tested yet. That is only was examined in Arabidopsis plants, so-called. It is a model plant often used in genetic experiments in plants, but it wasn't on plants yet. It wasn't tested in the field yet. There is one other complication. It's a GMO, genetically modified plant, and we have to stay with the protected greenhouses and things like that. These experiments were only initiated, but I cannot report yet the results. But it will go there for sure.
Okay. Interesting. So, what other uses do you envision? What about the efficiency of the photosynthetic efficiency? Do you think that this will increase that? Because I've heard, around the world, the average is like 1% for most plants.
It will raise the crop production. The extent would depend where you are and what light intensities you're exposed to and which plant you're talking about. So I won't draw a number in this table as yet. It is too early to tell. It is lab experiments and field experiments do look different in many cases. So I would like to be cautious on that. But from lab experiment, it does look very good. So this is one aspect I'm interested in, and I would like to talk to
Okay.
you about the languages that the organisms are using, because this is quite exciting, I think, as well.
Yeah, what do you mean the languages that they're using? Tell me about that.
Yeah. It is, you look at the drop of water, or you look at the lake, you look at the pool, whatever you look at, there are quite a number of different organisms there, and every once in a while somebody is taking over and you get what we name a bloom. This is a large rise in the population, a large change in the dynamic of the population. Now, the organisms excrete a large number of secondary metabolites. What are secondary metabolites? Those are the metabolites that are not part of the main biochemical pathways, namely carbon metabolism, nitrogen metabolism, forming proteins, etc., etc. But those are sidetracks, kind of. But if you take a cyanobacteria in a bloom, a blue-green alga in a bloom, and those intensified quite severely over the last decade or so, for a reason not really understood. We can go back to it, and if you wish. But they produce a large array of secondary metabolites. Some of them are toxic. You may have heard about toxic cyanobacteria blooms in Ohio, in Florida, along the Mississippi River. The beaches are closed, people are warned. The Erie Lake, one of the large lakes in Toledo City, got disconnected from water supply, not to mention the situation in China and other places. So the question is, in my view, first, is what is the biological role of these secondary metabolites that are some of which are toxic? And for instance, one of them is a so-called cylindrospermopsin, never mind the name. And it turned out that the organisms that produce it excrete it to the water. And what they do, what the metabolite does, is induce the formation and excretion of a protein that cleave phosphate from organic materials, and consequently, the organism is able to use phosphate, which is quite often a limiting nutrient in the water body. And the students that did the work named the paper enslavement in the water body, because the organisms that produce the cylindrospermopsin thereby raise the supply of phosphate because it enslaves other organisms to produce the alkaline phosphatase, which is the protein that cleaves the phosphate, and then it can grab it from the water quite efficiently. So this is one of the languages.
What organism compels or enslaves another organism? Which organisms are involved? Is this plants or is this algae or what?
Yeah, it is a blue-green alga. It's cyanobacteria. It is a filamentous cyanobacteria, so-called phanizomenon, there was a very, very strong bloom of that in the Sea of Galilee back in 1994, when I got a phone call from the Ministry of Science in Israel if I will be willing to look into it, and ever since I'm playing with it. I'll put it this way. Organisms from yeast to bacteria, when they face a phosphate limitation, they activate a regulon, a bench of genes, so-called 4-regulon, with one of the genes there encode a alkaline phosphatase. This alkaline phosphatase is excreted to the water and cleave phosphate from organic substances, making it more available for the phytoplankton. So rather than excreting a large amount of proteins, what this phanizomenon does is form cylindrospermopsin. The price of making it is much smaller than making alkaline phosphatase. Excrete the cylindrospermopsin to the water, and other organisms such as green alga, for instance, like Chlamydomonas, start producing alkaline phosphatase and excreting it, even though it has ample supply of phosphate. It is not limited by phosphate, but the cylindrospermopsin induces the formation of this alkaline phosphatase. Was I clear? Or?
Yeah, that's more clear. What do you think is the reasoning? What is it signaling in the the slave organism that it needs to create abundance of this? What do you think it's doing?
Yeah, if you follow, we know the sequence of events, and I won't go into the exact molecular sequence and network. But at the end of the day, if you add cylindrospermopsin to a culture of Chlamydomonas or many other green alga or other cyanobacteria as well, and you follow the gene expression pattern, there is it is sensed as a stress, as a phosphate stress, and activate the 4-regulon in that Chlamydomonas, for instance, and excrete alkaline phosphate, despite the fact there is ample of phosphate outside. So it is a signaling molecule that the organism sense as a signal. For instance, another toxin, mycocystin, which is one of the worst toxins in the water bodies. It has two biological roles. One of them is telling the organisms that the population is under oxidative stress, and it activates the formation of mycocystin within the mycocystis population. Or maybe I'll put it more clearly. Mycocystin is formed by mycocystis cells. These are a cyanobacterium, a blue-green alga. When some of the cells lies due to a stress, mostly oxidative stress, the appearance of mycocystin from outside the cell is sensed by the other cells, by the cells that experience the mycocystin in the environment, to activate a stress response. One of the pathways is large induction of the genes that form the mycocystin itself. So it raises the level of mycocystins. Now, the other biological role of mycocystin is to bind to protein and protect them against oxidative stress. Now, this is just another dogma that was turned wrong for quite a number of years. People were claiming that this toxin, mycocystin, is evolved as a way against predation. But the phenological analysis clearly indicated that it evolved, it is with us well before the eukaryote cells, well before the predators.
So how can this sensing go on from one organism to outside itself to determine that, oh, okay, if I release this compound, the other organism will take it up and do something beneficial for me? Like, how could there be that knowledge that this will work?
Within the population, it binds to a certain protein at the surface of the cell, and that signals the stress inward. Now, oxidative stress plays a major role in their daily life, I would say, and can also be used to combat these blooms. But this is a different story. We can go into it if you're interested in. We can use the oxidative stress, which
that'll be great, because algal blooms are a big problem. So let me know your thoughts on how this can be used.
Yeah, that is I wasn't going to talk about it, but a student of mine Dr. Moshe Harael developed a fascinating system there. The point here, we do have this serious problem of blooms, and what people are doing is using a huge amount of either copper salts or hydrogen peroxides, both with impact, severe impact on the ecology. Now, what Moshe Harael does, he formed a company that, well, I suppose we won't go into that commercialized aspect, but the biological aspect. What you do there is you induce the oxidative stress, and consequently, the organism, if you do it correctly, the organism enter a so-called programmed cell death. I'll give you an example. We teach the students that if an organism acquired ability to, for instance, detoxify hydrogen peroxide, it can fight back and it can survive well, and it has advantage under oxidative stress. But this is terribly wrong. If you take a chlamydomonas cell, for instance, and you want to kill the cell by hydrogen peroxide, it would take something between 5 and 10 millimolar of hydrogen peroxide to kill the chlamydomonas cell. But if you take quarter of a millimolar, you treat it with a quarter of a millimolar and come back after 4 to 6 hours with another quarter or half a millimolar, altogether less than 1 millimolar, at least 50% of the cells will die activating a programmed cell death. Programmed cell death is a genetically controlled mechanism whereby the cell altruistically kills itself. There are quite a number of examples there during development. If you look at your fingers, for instance, the skin that was between your fingers was perished because of activation of a programmed cell death. And therefore you have your fingers, not like whatever.
Webbing.
So if you, what Moshe Harael did was to place the peroxide, encapsulate it in hydrophobic component, and consequently it floats at the surface of the water, releasing the hydrogen peroxide very, very slowly. So when the mycocystis, for instance, or other blue-green algae are exposed for a longer time to a very low level of peroxide, what you get is activation of the PCD. And consequently, the population dies out not directly by a little dose of hydrogen peroxide, but using a much lower concentration, a sublethal concentration that activate the death. So it can come to the water body and treat it with a very, very low level of either copper sulfate that does the same, or hydrogen peroxide, and eliminate the cyanobacteria, which are far more sensitive to oxidative stress than green algae. I can explain to you why. There is a very good reason for that cyanobacteria is more sensitive to oxidative stress than green algae, for instance.
Well, I would think there probably would be unintended effects. How long would you need to put this low level of peroxide into a lake or an area that has an algal bloom, and what would be the secondary effects?
You see the effect within less than 24 hours. He did it in quite a number of places by now, in a lake in Ohio, in several places in China. The beauty there is that the other algae are not affected. And because of what I mentioned, it's because of the very low dose that is being used for a longer duration. And as a result, if you follow, for instance, the chlorophyll level decays a bit because of the death of the cyanobacteria, but the green algae that are there are taking over. And now they compete with the cyanobacteria. And for instance, a lake in Ohio treated, if I'm not mistaken, sometime end of July. There was no bloom anymore for the rest of the season. And people were quite happy about it because for the first time in a few years, they could jump into the water and consume that water. So it is a very novel, very clever way to avoid the ecological impact of the high toxic, such as copper, high dose of copper, and hydrogen peroxide. With copper, what people did was so far is, since the copper grain is much its specific gravity is way over that of water, it just sinks to the bottom. But by encapsulating it, it floats on the water, even move with the wind, with the scums of the cyanobacteria. Quite clever way of doing it, and
that's excellent. So, maybe last question for now. Why is the cyanobacteria more or less resistant to this than the other forms of life?
Yeah, well, they are more sensitive to peroxide. There is a very good reason for that. It is if you take higher plants or green algae, and there is excess electrons coming from photosystem after absorbing light energy, and there is not enough CO2 to fix at the end of the path. So they are CO2 limited. A plant with underwater stress closes its stomates, and it is limited with CO2 supply. And it is a matter of the donor and the acceptor. The photosynthesis, the reaction center releases many more electrons that can be used to reduce CO2, which is not there. So they go to oxygen. In higher plants and green algae, it is so-called the Maillard reaction, the name of Maillard. It forms hydrogen peroxide. So evolutionary speaking, quite a number of different mechanisms evolved in green algae and higher plants to detoxify the hydrogen peroxide, the singlet oxygen, the superoxide, and the hydrogen peroxide at the end of the day. In cyanobacteria, they have a different way of transferring the electrons to oxygen. A student of mine, Yael Hellman, discovered it back in 2003. They use a protein so-called FLV. It is never mind the exact name. These proteins take up the 4 electrons of coming from the reaction center and convert it to oxygen in water directly without releasing active oxygen species, such as what I mentioned, superoxide, dismutase. Activities are not necessary there. Hydrogen peroxide is not formed. So evolutionary speaking, the cyanobacteria are limited in their ability to detoxify hydrogen peroxide, and therefore they are more sensitive. So you can use a very low level of hydrogen peroxide to kill them, much lower than needed, for instance, in algae.
Well, very good. Aaron, we're almost out of time. What's next in terms of your studies and your student studies? Now that you've seen some of these mechanisms, what's next for you to look at?
I'll say two different things. The one thing I'm really, really proud about is the very large number of students of mine that got independent academic research positions. And I'm collaborating with quite a number of them still. The one thing to do is to gain a better understanding of the metabolic flexibility that the chlorella that I mentioned before is using in order to be able to grow that fast. Because one of the main questions here is what sets the ceiling on the growth rate of a photosynthetic organism. This is the main question that I'm dealing with quite these days. The second question, and actually the mission, I think, is to get rid of these toxic blooms, to reestablish, or if you wish, to reset the ecological health of the lakes. It is. We need to I would put it this way, to make the Chinese people reuse the lakes water, get rid of the bottled water that they use, and reset the lakes. If you know, I just came, well, luckily about 2 months ago, before the coronavirus, I visited China. It's terrible there. I was there for quite a number of times. Really? It's terrible there. The lakes are contaminated with blue-green alga, and they don't know what to do about it. It is true that the organisms depend on the nutrients, and there is excess of nutrients, and they won't grow without the nutrients. But it would take hundreds of years to get rid of the nutrients in these lakes, and the Chinese people do need the solution the day before yesterday. So if we can come up with a good way of doing it, and I think we do, that will help a lot.
Well, Aaron, thank you for coming. I know you're speaking to me from Israel, and not from a major city area. So I appreciate you're looking into this stuff. Some people would think it's obscure, but it's very important, and it has very important consequences. So I appreciate you being on the podcast.
Yeah, thanks for this discussion this evening. It was fun talking to you, and I hope it helped a lot to people to understand where we are, both in the desertification aspect and
for people listening. How can they? You said you have many students. How can they find out if they have a problem in their area with algal blooms? How can they read papers or get in touch with you or your students to learn more?
I find it is. I really think it's our mission to get rid of these blooms, and I'll be willing, if they don't flood me too much, I'll be willing to talk to people, to answer emails, and help as much as I can. Because, I'm facing quite a number of cases where people pay awful lot of money for nothing. And
for solutions that don't work?
No, the solutions don't work, and the promises are without any basis. Yeah. And that makes me furious about it, really. It is unbelievable what people do. I don't think it is very surprising, but this is I wasn't aware of it before, really.
Well, very good.
Forget frequently asked questions, common sense, common knowledge, or Google. How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5% go above and beyond and become very good at what they do. But only 0.1% are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in fields such as sleep science, cancer, stem cells, ketogenic diets, and more. Get ready. Here come the geniuses. This is the Finding Genius Podcast with Richard Jacobs.